So there are many multiple sequence alignment softwares, programs. There is one online program which is most famous. So I will go with you guys with this Clustal W, but feel free to use rest of them depending upon what research questions you are after. So this one is called Clustal W. It's cluster analysis and then a weighted scheme where we can do some clustering, we can do some make some trees and give some weights to those aligned sequences. It's a progressive global multiple alignment program, the web link, and presented by Thompson et al. In nucleic acid research in 1994. There are other variants also. There is Clustal X and Clustal Omega, which are also most frequently used. So it performs pairwise alignments and use the alignment scores to produce the phylogenetic tree, and then sequentially align those trees guided by the tree. So that is what we have learned in the progressive methods. So the most closely related sequences are aligned first, then additional sequences are added. The initial alignments used to produce the guide tree may be obtained by a fast k-tuple approach or slower dynamic programming methods. We have seen this k-tuple approach when we were doing those FASTA and BLAST searches, where we see that you can take the whole sequences and you can divide the sequences in different tuples, different chunks. So that approach we can use it over here also. For building a tree, genetic distances between sequences are calculated as the number of mismatch positions in an alignment divided by the total number of match positions. So mismatch divided by match. So this is how there are more mismatches, more sequences are distantly apart. There is more distance in them. There are if this number is a small number, obviously the sequences are more closely related. We will use those distance methods in our phylogenetic lectures. So we come up with those trees where we put all those sequences. So for example, here we have three sequences A, B and C, and they are put together. And if we look into these tree, we can see that the overall tree is divided into two parts, where we can see there is a C, we call it as if some sequence is present separately from rest, so we call it as an outgroup. And this tree has a root also, so we call it as a rooted tree, but I will repeat these things when we are dealing with the those phylogenetics stuff. So we take those scores. So we have for A we have 0.2, and then since this is sharing the node with which is coming from 0.3, so in this way we have equal weightage for all of the sequences. So this 0.3 by 2 is equal to 0.35, and then this 0.35, since it's coming from here, we are getting 0.5 towards top, 0.5 towards bottom, so it's divided by 0.5, and in this way the overall weight for this particular sequence A is 0.7. Same way we came up for B we came up with 0.5, and for C we came up with 1. So in this way these individual sequences are normalized with some weights, with some weighting factors, and this weighting factor actually normalizes these sequences on the tree. We use those weights, and then we would see which ones go with each other. So for example, here we have one column in which we are focusing. We have K, L, I and L. Sequence A, we use its weight A. Sequence C, we use weight C, and then B and C. So we take one alignment, and then we see what are the most appropriate matching of these nucleotides. So that is we can get with this relation down there. So here is the page for this Clustal W. In fact, its latest version is Clustal W2. What you can do over here is you will find a space, in fact a big text area where you can put this your sequences. So for the practice, I put Arabidopsis genes, my favorite constans genes family. which I worked a lot. So we took those genes, actually the homologs. There is when we have most of the genes homologs with one another in an individual, we can classify them as gene families. So I put this constants genes family. There were those genes which are belonging to this constants genes family. I put them in this, and then I let it go. Before we go, we check some options. So here there were some default options. You can play and you can change them. In the default, we see that the Blossom matrix is used. Gap open penalty is 10. Gap extension is 0.20. Then gap distance is no end gaps. I let all the defaults checked. In clustering methods, it says neighbor joining. That's where we read them in our phylogenetic lectures. And then we check this or click this submit button. So it brings up this alignment like this. So we have the residues which are aligned. We can see those matches and mismatches in different colors. Where we have those sequences conserved, we will see the sterics down there, and those all of them are the same colors. And for 20 amino acids, we have 20 different colors. So where we can see colors are changing in the columns, obviously we find those mismatches over there. Then it makes a tree, a guided tree. We see that the option in this radio button, which is checked as cladogram. So in cladogram, we see that all those sequences are we can see their names which are present on the equal distances. So we can see they are kind of a symmetry in this tree. But if we check the real one, then it can bring their real distances into it, and you can see some of those lines are shorter and some of them are longer. So in this way you can incorporate their actual distances from one another. There are many options. You can play with it. We'll design some simple homework where you will play this cluster W, and I know that you will use it a lot if you're doing some biological research. So we see it's a progressive global multiple sequence alignment program, cluster W, which is the most famous multiple sequence alignment program used.